Hello, welcome to my poster presentation. Our topic will be basal blood pressure characterization of smooth muscle-specific IL-36R knockout mice. To provide you with an overview of our research, high blood pressure or hypertension is a major factor in premature death worldwide, and its underlying mechanisms remain unknown. Preliminary studies show that an association of hypertension with an increased expression of interleukin-36 receptor, or IL-36R, which is an inflammatory regulator, is in aortic vascular smooth muscle cells, or otherwise known as VSMCs. Therefore, we are investigating if reduced IL-36R in VSMCs will be therapeutically beneficial in preventing hypertension. To do this, we developed genetic IL-36R knockout mice and characterized the blood pressure in these mice at basal levels without stimulation. For knockout mice, IL-36R flox mice were cross-mated with SM22 Cree mice, and their offspring were then identified by PCR and genotyping. Once confirmed, non-invasive tail cuff blood pressure was performed on IL-36R heterozygous knockout mice and their genetic control mouse lines, including IL-36R flox, SM22 Cree, and wild-type mice, which are represented as WT. Results show that there were no significant difference in measured systolic and diastolic blood pressures, as well as the calculated mean arterial pressure between IL-36R heterozygous knockout mice and the control mice at 3 months of age, meaning that partial reduction of IL-36R in vascular smooth muscle cells does not alter systolic, diastolic, or the MAP. This provides our baseline data to investigate knocking out vascular IL-36R and angiotensin II-induced hypertension mouse models to validate whether IL-36R mediates hypertension under pathological conditions. To gain our results, we used genotyping, a blood pressure measurement system, and statistical analysis. For genotyping, small pieces of tail tissue were collected and DNA extraction was performed. DNA samples were added into the PCR reaction mixtures that contained red extract, DECP water, and primers. IL-36R and SM22 Cree PCR reaction mixtures were ran at different thermocycler conditions as shown. PCR products from each sample were then inserted into a 1.2% agarose gel and ran at 235 voltage in TEA buffer, at which the gel plate was reviewed under the UV light to visualize the target bands. Our blood pressure parameters were measured using the Kent-coded non-invasive blood pressure machine, which can be seen here. Occlusion cuff and VPR cuff were attached to the channels. Mice were placed into the holders with the nose cone and a rear covering that allows their tails to be seen out of the holder for measurement, and you are able to visualize it here. Lastly, our results were presented in an average of plus or minus standard error. One-way ANOVA was used for statistical analysis, and the p-value greater than 0.05 as significant. After data was completed and analyzed, genotyping results confirmed SM22 Cree, IL-36 flox, IL-36R flox crossed with SM22 Cree, and wild-type mice. These two examples highlight some of the genotyping that was performed for this research. They show positive transgene band for the SM22 Cree and the mutant band of IL-36R in crossed mice. If you look here, you can see the latter for reference to see our band pairs. You can also see our genotype numbers 16, 17, and 18, along with our positive controls located here, and our wild-type controls here, and our water control. Now to shift focus to our blood pressure data, a mouse's normal blood pressure range varies between strain, but it is typically around a 122 for the systolic, 102 for the diastolic, and 112 for the mean arterial pressure. Our mice averages exhibited no significant difference between crossed mice and controlled mice at 3 months of age. Blood pressure averages were also lower in male knockout mice than their female counterparts. And if you take a look over here, you will see our graphs. So we have systolic, diastolic, and our mean arterial pressure. It is measured in millimeters per mercury, and our genotypes are listed as wild-type IL-36R, SM22 Cree, and our crossed mice. You'll also notice our p-values here, and they are all above 0.05, showing that they are not significant. Moving forward, we intend to induce hypertension in heterozygous knockout mice by angiotensin II infusion to determine whether partial reduction of IL-36R prevents induced hypertension. We will also do the same for homozygous IL-36R knockout mice to determine whether deletion of IL-36R will protect against hypertension as well. I would now like to thank my lab mates, mentors, and Dr. Q at Center for Translational and Molecular Medicine Lab at Georgia State University, as well as the Honors College here, and the funding provided by Grant HL115195. And again, thank you for listening, and I hope you enjoyed my presentation.